name's Arun. I'm a postdoc now with Jody Hay at Temple University. And I'm going to tell you a little bit about, we've heard a lot about reproductive incompatibilities and introgression, and how we've been estimating them, or trying to estimate these using summary statistics. Here's a different approach, and I'll tell you why this is a much more robust approach to doing that. So I'm going to start off with something really broad. When we think about what happens when populations diverge and start to exchange or continue to exchange genes over evolutionary time, and you think about some sort of a traditional isolation with migration model, we've got an ancestral population here which splits off into two daughter populations, and they continue to exchange genes over evolutionary time. What we've come to know over through theoretical studies as well as empirical studies is that not all loci migrate equally. I know that sounds horrible, but it's true. And this kind of leads into a lot of speciation theory when we think about what contributes to these so-called genomic islands of speciation, which have been, again, found in multiple species, which have been thought to traditionally comprise elements which lead to hybrid incompatibilities and reproductive isolation over evolutionary time. And when we think about what this looks like at the genomic level, this is a scan. Traditional, you see a lot of these scans. We'll see a lot of them through talks here. If you think about what we're actually looking for, we're looking for these outliers, right? So we're thinking, across the genome, we see some sort of nominal level of FSD, for instance, here, or DXY, or DA, or pi, G minus D, whatever you're looking for, you name it. What you're really looking for are these outlier loci, something that sticks out like a sore thumb, and you're going to say, all right, we're going to predict that indeed there might be something going on evolutionarily which is contributing to this increased differentiation at this one particular locus. Right? So you see that in a lot of literature, and a lot of literature also calls these as genomic islands of speciation, for instance, just because they happen to be at loci which might contribute towards reproductive isolation. These have been found in lots of species. A lot of the species, studies are also amongst us right now, from flycatchers to sunflowers to butterflies and so on and so forth. But the problem here, as rightfully pointed out by this very seminal paper, I love this paper by the way, I think I see Matt on back there. Good job. Don't look back. So the problem at this kind of studies using FSD or DXY or summary statistics is that what you're really looking at are these regions of high differentiation. What you're really looking at or capturing at these loci are reductions in effective population size or reductions in diversity. And this could be due to multiple reasons, all right? So this could be due to, yes, indeed, due to differential introgression or positive selection, negative selection against migrant alleles between two populations. Or it could be due to sweeps and divergence hitchhiking, which is basically just linked natural selection at neutral sites, because there's a sweep going on somewhere else, you have reduction in diversity at neutral sites just by virtue of that. And so they say this in their paper, and it's true across a lot of studies now that we've looked at, that there's really no sort of consensus on what kind of statistics that you really want to use to determine signatures of differential introgression or link selection across your genome. And we were going to take so that's the problem, that's the heart of the problem here. How do we actually decouple the effects of link selection and differential introgression at genomic loci, all right? And we're going to try and address this, all right, we've seen summary statistics so far, we're going to address this using some model-based methods now, all right? A part of this solution was proposed by this very nice paper by Vitor Sousa and colleagues. Vitor was a postdoc with Jody as well a couple of years ago. And he proposed this Bayesian MCMC method, sort of in addition to the IM suite of tools, if you're familiar with them, the isolation with migration suite of tools. What they're ultimately doing is a Bayesian MCMC formulation to approximate the posterior density of evolutionary parameters, in this case, effective population sizes, migration. What happened? Oh no. I guess they don't want to hear me talk. No, I'll just go on. Okay, can you guys see the slides though? I have all white background, so you should be all right. I'm just going to keep going. All right, so the idea here is that Vitor proposed that, all right, we're going to have, say a couple of hundred loci that you're looking at. Let's split these out, and now we're out. Anyway, that's going to make a fun video back there. It's just videotaping my talk right there. All right, so let's split up these loci into two groups, okay? And let's say one group of loci are migrating or have lower migration, for instance, and another group has higher migration, all right? Or you can do the same thing with groups of loci for effective population sizes, for instance. And let's say one group has lower effective population size, the other one has higher. Okay? And not to go into too many details, it uses an MCMC to sample from posterior densities. It integrates over genealogies and assignment here in terms of assignment to these groups of loci. Okay? And they also proposed this nice likelihood ratio test similar to Nielsen 2007 to test nested models against different models of evolution here. All right, so we're going to propose a part 2 to the solution. All right, so I'm going to tell you about 3 different tests that we came up with, which can actually help us decouple the effects of differential introgression and link selection. Okay, the first one is the likelihood ratio test of differential introgression. The second one is one specific for link selection effects. The third one is a test of independence between these two. Okay. So the first one is a very simple, just if you look at this cartoon here, under the null, okay, you would expect to see all loci that you're analyzing to contain some sort of a unimodal distribution for posterior density of, say, the migration rates here. Okay. So essentially all loci that you're analyzing seem to be migrating the same way at similar migration rates across this model. Okay. But the alternate hypothesis, indeed, is that you've got one group of loci which carry some sort of a low migration rate indicated by these thinner arrows here, as against another group of loci which are indicated here by these thicker arrows, which seem to have some sort of a higher migration rate. And so the alternate hypothesis, I mean, if your alternate hypothesis is indeed true, you would expect to see, all right, some bunch of loci kind of sticking out here, and then you can already, you can sort of reject the null and say, all right, these loci are indeed migrating. The second test is very similar. In this case, we're testing for link selection effects. So what you're looking for are reductions in diversity, reductions in effective population size. And so what you're seeing here under the null is that you've got one sort of unimodal distribution for posterior density of population sizes. Whereas under the alternate hypothesis, you're going to see one group of loci which show squeezed up population sizes under this model here, whereas the other group seems to show higher population sizes. So that's kind of what you'll expect to see under the alternate. And then the third test is a very simple test. It's a test of independence between these two loci. All right, now that you know assignment of loci into groups of differentially introgressing loci, groups of loci which are probably under link selection effects, all right, now you've got a simple Fisher's exact test of independence between these loci. Under the null, that these loci affected by link selection are independent of loci that are affected by differential integration. Okay, and the alternate is that they're not. Okay, so that's a simple contingency table that we just draw up. Okay, so I developed a new method. So I took Vitor's method and I parallelized it. I wrote a new software for this. I summarized a year's worth of work into just saying it's pretty cool because it's very fast. You can analyze lots of loci as against spending years running IMA2 doing the same thing. I actually reviewed a paper which took a year and a half to do IM analyses. I'm like, what? Okay, so it's parallelized now, which is cool. So you throw a number of, lots of processors at it and it runs fast. It's great. And what I did was I curated a lot of species pairs here. So I got 8 different species pairs from a lot of different studies here, which vary in natural histories, amounts of known migration levels between these populations, modes of speciation, different types of genomic data, etc. A couple of these are not published yet, so anyway, I'll tell you a little bit about that. So that's the Drosophila set here. But they're all over the place. So we got Uriculagus, which is European rabbits. You got sticklebacks. You got a couple of pan data sets. So these are chimpanzees and bonobos. Then you got Drosophila, Pseudoobscura, and Priscilla. And for all the genomic data sets that I curated, I pulled out 200 loci. I'm going to show you some results. So this set of results is just a simple IM analysis. Okay, so first shot at it, we took it. We wanted to see if there are actually signatures of migration that we see, significant signatures of migration across these species pairs. And indeed, what we see are some pretty cool signatures here. Lots of bidirectional migration rates between the heliconius butterflies, these rabbits, the anopheles. This one's the bonobos into central chimpanzees. This one's the central into western chimpanzees and Drosophila. A couple of these reject any significant migration. Now these are results from the first test that we did, and so these are tests of differential introgression here. And what we see is that indeed a lot of these species pairs reject the null and accept the alternate hypothesis that there are indeed two groups of loci that we analyzed which seem to be showing signatures of differential introgression across the loci. Okay? So that's Heliconius, this one's the chimpanzee, Anopheles, Drosophila, whereas the sticklebacks and mice and bonobos reject that. Okay? And then if you look at the second test, so this one's a test for link selection effects, we got a bunch of species, the ones that actually rejected differential introgression stick out here. They showed signatures of link selection effects. And the ones that I'm showing in blue are the ones that also had signatures for differential introgression in the previous set of tests. Okay? And then a couple of them rejected, which is very cool. So now you can actually take these three species pairs and do a test of independence, which I talked about. And we can actually see here, we reject a test of independence across two of these species pairs, so that's Anopheles and Drosophila, which is very cool. So now you have, an actual test that you can do to determine to see if there's link selection or differential introgression effects, which is very nice. And if you look at the actual posterior density distributions, what they look like, they look very similar to those cartoons that I showed you. So you got one group of loci sticking out closer to 0 population sizes out here, another group of loci shown here which have much higher population sizes. If you look at the density distributions for the migration rates, you see similar patterns. You got one peak up here and then a flat distribution down here. And you see that across all the species pairs that tested which had positive differential introgression or link selection effects. So in conclusion, I'm going to just summarize this. So we found lots of very interesting patterns. We got a couple of species which have both differential introgression and link selection effects. And ultimately what we would love to see are all what you folks work on, which is want to see if there are actually functional elements present across these loci which are indeed leading to effects of differential introgression or link selection. And yeah, the software should be out on my website sometime soon enough. So with that, I think I'm done. I'd like to thank Reacher. Reacher is at Bern now. A lot of really nice folks who were nice to share their data with us. The NIH grant for funding, as well as Temple is something as a grant, and everybody in the HALAB. With that, another shameless plug. I'm starting a faculty position at Cal State San Marcos this fall, so if you're interested in my work, I'm looking for postdocs and master's students, come chat with me. All right, I'll take questions.